properties.
Visual processing begins in the retina, the receptive surface inside the back of the eye. Light enters the eye, passes through the layers of cells in the retina before reaching the photoreceptors, which are located at the back of the retina. Light activates the photoreceptors, which modulate the activity of bipolar cells. Bipolar cells, in turn, connect with ganglion cells located at the front of the retina. The axons of the ganglion cells form the optic nerve, which carries information to the brain. Two other types of neurons, horizontal cells and amacrine cells, are primarily responsible for lateral interactions within the retina. The bipolar cells and ganglion cells are organized in such a way that each cell responds to a small circular patch of photoreceptors, which defines the cell's receptive field. The receptive fields of retinal ganglion cells are concentric, consisting of a roughly circular central area and a surrounding ring. Retinal ganglion cells have two basic types of receptive fields: on-center off-surround and off-center on-surround. The center and its surround are always antagonistic and tend to cancel each other's activity. Let's look at the response of an on-center ganglion cell to a spot of light. When no light is falling on the receptive field, a spontaneous level of activity is recorded from the ganglion cell. Notice that when the light enters the surround region on this on-center ganglion cell, the level of activity recorded in the cell decreases. Conversely, a spot of light in the center of the receptive field increases the firing rate. A maximal response in an on-center ganglion cell is achieved when the entire center of the receptive field is illuminated. Likewise, if we illuminate only the surround with a ring of light, the ganglion cell is maximally inhibited. Note that if we illuminate both the center and surround region, the response is just above baseline. This occurs because center effects are slightly stronger than surround. Now let's look at the response of an off-center ganglion cell. Notice that when the light enters the surround region of this off-center ganglion cell, the level of activity recorded in the cell increases. Conversely, a spot of light in the center of the receptive field decreases the firing rate. If we illuminate the entire center of an off-center ganglion cell, the cell is maximally inhibited. A maximal response is achieved when the entire surround of the receptive field is illuminated. As with the on-center ganglion cell, if we illuminate both the center and surround region, the response changes very little from baseline. As you can see, uniform illumination of the visual field is less effective in activating a ganglion cell than is a well-placed small spot or a line or edge passing through the center of the cell's receptive field. This organization makes the ganglion cells sensitive to differences in the level of illumination across the receptive field, or what is called luminance contrast. To understand the importance of luminance contrast, let's see what happens at the boundary between light and dark regions. Note how the response rate changes depending on the position of the receptive field. By combining information from adjacent receptive fields, the brain can thus construct information about edges and ultimately shapes. This graph shows the responses of a hypothetical population of on-center ganglion cells whose receptive fields, labeled I to V, are distributed across a light-dark edge. Those cells whose activity is most affected have receptive fields whose center region lies adjacent to the light-dark edge. Thus, the information supplied by the retina to the brain does not give equal weight to all parts of the visual scene. Rather, it emphasizes features of the visual scene, such as boundaries and edges, that convey the most important information.